guys, my name's Katherine. I'm one of the tutors here at Chegg. I typically tutor science, English, some math, and Spanish. And today we are going to be talking about a topic in science, specifically biology and chemistry, and that is the acid-base balance in your blood. So the first thing we're going to talk about is a quick overview of what acids and bases are. So we have this molecule here. Let's call it HX. And in this case, X is just anything that's attached to a hydrogen. Sometimes when chemists use an X, they mean that it's a halide, which is a specific type of element, but here we just mean it's anything at all. So here we have HX, and then it's in an aqueous solution, which means it's dissolved in water, and then it can break up into an H plus and an X minus, also in this aqueous solution. And the opposite can also happen. These two things together, floating around in water, can come together and they can make HX. And the strength of these two reactions, this one is called the forward reaction from HX to H plus and X minus, and the reverse reaction from H plus and X minus back to HX. Depending on which one of those two is stronger is a measure of whether it's a stronger acid or base. So if this forward reaction is the stronger one, then we would call HX an acid. If we were to go the opposite way, we would call X minus a base. And in fact, X minus is what's called the conjugate base of HX. Or you could say that HX is the conjugate acid of X minus. And another way to think about bases, instead of thinking them as things that pick up hydrogens like this X minus does, we can also think of them as things that donate OH groups. So for example, we have here this XOH, where once again X is just any molecule at all. And it donates an OH group in this aqueous solution. So it breaks up into X plus and OH minus. But what can happen in this situation is that this, let's make that black again, but this OH minus here, well, let's make it green for seeing better. So this OH minus here, and this H plus here, come together, and we can make H2O, which is water. And water has a pH of 7. It's equally likely to donate a hydrogen by making an H plus and then it makes an OH minus at the same time. So the pH of pure water is always 7. And so by adding this, if we just had this thing, which is acidic, like we said before, that's going to bring the pH down, which means it's more acidic. And then when we add this, it brings the pH up, which means that it is basic. But even so, with this reaction right here, say if this reverse reaction were even stronger, then we would say that that's a really strong acid. It's taking up OH minuses and leaving a lot of H pluses. So the point being that every acid has a conjugate base, every base has a conjugate acid. And in order to get the right pH, we have to add hydrogens or OH groups in one way or another in order to get there. So when we're in the lab, that's relatively straightforward. We just decide the pH that we want to get to. We have machines that we can use to test, and we can just keep adding acids and bases until we get to the number that we want. But that's not the case in the human body. We can't just keep adding H pluses if we need a solution to be more acidic. So we need another way to do it, and we need a way to do it really, really well, because a lot of the things in the body, a lot of the proteins need a very specific pH value in order to do their job well. So for example, the pH of blood should stay at 7.4, which is just a little bit basic, because 7 is not acidic, not basic. 1 is super acidic. 14 is super basic when we're talking about pHs. So blood has to be just a tiny bit basic. And how we do that is with this really cool molecule right here, H2CO3. So if we have a very basic, or a little bit too basic of a blood pH right now, so say instead of 7.4, it's at 7.5. So it's a little too basic, and we want to make it more acidic. So how do we do that? We need to add more hydrogen. So this molecule right here that I just mentioned, H2CO3, can break up like this. So we'll have an H plus, and then an HCO3 minus. So this is called acidosis. It makes the blood more acidic to bring the pH back down to 7.4. And that's, exactly what we need. But say now, next day, something else happens, and now your pH is at 7.35 or something like that. So now it's a little bit too acidic. What are we going to do? So we do the opposite reaction. So it's not just that we have a bunch of this sitting around, we also have some of the byproducts over here. And as the pH gets a little bit too acidic, a little bit too acidic, some of the H pluses and the HCO3 minuses form into H2CO3, decreasing the number of H pluses in the solution, and thus making it more, decreasing H pluses, thus making it more basic. So the pH would go back up to like 7.4, and it's a balancing act. Another thing that H2CO3 can do, in fact, instead of following this acid-base reaction, it can follow a non-acid-base reaction, which we see on the right, where it makes liquid water and CO2. This is important for other processes that the body goes through, but I just wanted to include it there just in case you were thinking like, well, what if it did this thing instead? Yeah, it definitely can do this. It's just not super related to acid-base problems. One last thing, in case I didn't say it before, I mentioned that this first process here, that's called acidosis. The reverse process down here, that's called alkalosis. It's just another word for saying making something more basic. And so that will do it for the acid-base balance in your blood. I hope you enjoyed this lesson. And if you have any questions, I'm always available for private tutoring.